Hey, welcome to the Biochemistry Academy. We began a serial course on cell biology, and in our previous video, we talked about the cell cycle and the regulation of the cell cycle. Well, in today's video, we'll talk about the M phase of the cell cycle, which is mitosis and cytokinesis. So let's do a brief review of the cell cycle. We said the cell cycle is divided into two phases, which is the interphase and the M phase. And the interphase is comprised of three subphases, which are the G1 phase, and this is where cells grow, and the S phase, it is where there is synthesis of the DNA, and the G2 phase, this is where the cell grows the more and prepare itself for the next phase, which is the M phase of the cell cycle. And the M phase represents cell division. The cell division includes mitosis, which is the division of the cell nucleus, and cytokinesis, which is the division of the cytoplasm. And remember, we said that cells spend most of their time in the interphase. Well, in our today's video, we'll talk more about the M phase of the cell cycle. Now, it is mitosis that brings about the growth of an organism and the repair of damaged tissue by producing identical body cells. So wherever you see the growth of an organism or the repair of a worn out tissue, it is brought about by mitosis because it produces identical body cell. So this mitosis is further broken into four phases, which are Prophase, Metaphase, Anaphase, and Telophase. We can use a simple mnemonic like PMAT to easily remember the phases that are in mitosis. Remember, we said that they are prophase, metaphase, anaphase, and telophase. So let us look at the first one, which is prophase. It is the longest phase of mitosis, and in this phase, chromatin begins to condense into the shape of chromosomes, and the nucleus disappears. Now, the previously coiled DNA coils tightly to form sister chromatids. Individual chromosomes are formed. Now, this is the structure of a chromosome here. And at the center of the chromosome is the centromere that attaches the sister chromatids together. And also in the prophase, from the cytoplasm, microtubules known as the spindle fibers begin to form from two sets of paired structure called centroles. The spindle fiber elongates as the centroles begins to move to the opposite sides or pole of the cell. While it's happening, the nuclear membrane surrounding the nucleus disappears. Then the opposite end of the spindle fiber can attach to centromeres. Next, it moves to the metaphase. In the metaphase, the centroles complete their movement to the poles of the cell, and then the spindle fibers align the chromosomes along the equator of the cell. This end-to-end alignment of chromosomes results in a sister chromatid on either side of the equator. Then it moves to the anaphase. In anaphase, the spindle fibers separate the sister chromatids at their centromere. Once they are separated, each chromatid is called a chromosome. Now, the single-stranded chromosome forms a V shape as the spindle fibers draws them through the gel like the cytoplasm, dragging the chromosomes to the opposite poles of the cell towards their centroles. Many a times students may confuse these centroles and centromere. Well, remember the centroles are at the poles, while the centromere is the center of the sister chromatid. So let's move to the next phase, which is the telophase. In telophase, which is the final stage of mitosis, in this phase, a nuclear membrane reforms around each set of chromosomes at the end. Then the chromosomes spread out into chromatin and the nucleus becomes visible again. So mitosis, which is the division of the nucleus, is now complete. Then next, which is the final step of the M phase, is cytokinesis, which is the splitting up of the cytoplasm. Now in animal cell, cytokinesis occurs through the inward movement of the cell membrane. This process progressively pinches the cytoplasm until two identical daughter cells are formed, while in the plant cell, it can't pinch the cytoplasm because of the rigid cell wall surrounding the cell membrane. Instead, the cell wall material assembles along the equator, forming a structure, the cell plate. Now, this cell plate grows until it joins with the existing cell membrane, separating it into two halves, which are the daughter cells. Over time, new cell wall begins to form between the two daughter cells. So note the key points to remember the M phase of the cell cycle. During the M phase, cell division occurs through two processes, which are 1. Mitosis, and we say that it is the division of the nucleus, and 2. Cytokinesis, which is the division of the cytoplasm. The mitosis have 4 phases, which are Prophase, Metaphase, Anaphase, and Telophase. In Prophase, chromatin condenses into chromosome, spindle fibers forms, the nucleus and the nuclear membrane disappears. Then in interphase, spindle fibers align the chromosomes along the cell equator. In anaphase, the spindle fibers separate the sister chromatids into two separate groups of chromosomes, pulling them towards the poles. And in telophase, the nucleus and the nuclear membrane reforms. The chromosome disperse into chromatin. Then in cytokinesis, which is the division of the cytoplasm, two daughter cells are formed. So the M phase is only completed after cytokinesis, resulting in two daughter cells which are identical to each other and identical to the original cell. Well, that's it for this video. In our next video, we will talk about meiosis. Please don't forget to like, share, and subscribe to the Biochemistry Academy for all biochemistry courses. Arigato.